Alright, good morning everybody. Sorry I'm not able to be there with you today. Christopher's homesick. He's got something bugging him, I'm not sure what. So I'm stuck at home today. So I want to take a few minutes. I want to walk you through the lab because I know when I went through this the first couple of times it confused the heck out of me. So I want to make sure we try to get it so that you guys aren't too confused while you're going through and working on this. Alright, so what today's lab is going to deal with is it's going to look at some of the factors that can have an impact on natural selection. What we're really going to be doing is getting you ready for starting to talk about the Hardy-Weinberg principle. And that's going to be our topic of discussion on Friday when you guys return from Chicago. By the way, sorry I didn't get to see you take off. Have a good time. I'm bummed I can't go. A lot of good biology going on there, but I won't go too geeky on you with that. So have a good time. Enjoy your senior trip. Anyways, you're going to be investigating something called the strawfish. And the strawfish is going to be made up of alleles. And just a reminder on alleles, what alleles are they're the factors that you inherit through genetics. You're going to use different straws to represent alleles. And so in my case, I have a red allele. I don't know if you can see this there. You know what? Let me switch to this. In my case, I have red alleles. And so the straw is going to represent red for me. And then I also have blue alleles. Alright, and remember, all traits are determined by a pair of genes, if not more than one pair of genes. Alright, and so what we're looking at here is these combined together are going to make up the genotype, and then the trait that you have as a result of this combination of genes is going to represent your phenotype. So just a little bit of a reminder from back in general bio. Then, additionally, we're going to be looking at the fact that the traits you're going to be working with today are going to be what we call incomplete dominance. So what that means is if I have two of the blue genes, or two of the blue alleles, it's going to have a certain phenotype. And in this case, we're going to say it's going to produce a blue fish. If I have two of the red ones, and forgive me if these are not both red, and if one of them's green, I apologize. You guys understand why that is. That's going to be a red fish. If I have the heterozygous version of it, I'm not going to classify that as dominant. We'll say blue is dominant in this case. It's not going to be considered dominant. It's going to instead have its own set of characteristics. So in this case, blue and red was, I think blue and red are purple, right? Being colorblind, I never really cared much about that stuff. But anyways, so then this is going to create a purple fish. Alright, so what you're going to do first is you're going to take your straws. You're going to get 10 of each color. Alright, and what you should notice in the classroom is there's a box of solid colored straws. There's a box of straws that look exactly like this as well. You're going to get 10 of these, and in groups of 2 or 3 working together, you're going to take those 10 and you're going to cut them in half, as close to being in half as possible. So you might want to get a ruler out so that now those 10 become 20. And I'm using fewer straws just to use as an example of this. So I'm going to take these ones, I'm going to cut them as well. So now I should have 20 of each color. You're going to take those 20 and you're going to put them inside the paper bag. 20, I got my paper bag. Now I'm ready to go. The house is messy behind me, I'm sorry. Alright? So then you're going to start working through a series of 4 different activities. Alright, in the first go around, what you're going to be doing is, well, I guess I should start by saying, you notice your phenotypes, there's 3 different colors. Make those colors match the colors that you actually have inside your bag here. So make the colors match. If you need to invent a new color because the 2 colors that you have don't actually go together to make a new color, have fun with that. Make yourself a new color and give it a creative name. Just keep it school appropriate. Alright, so then, first thing you got to do, in each test, you're going to do a control round first. We have to establish our population. So what I'm going to do is I need to figure out what groups are going to survive. So let me come back to this a second. Make sure you can see what's going on. So give me just a second to get the camera adjusted. Unfortunately, I can't zoom this out. I can zoom it out a little bit more. That's all the farther back I can go with it. Alright, so have this all the way back. Have all my straws in here ready to go. The first go around, I am not going to have any selection factors. So I'm just going to go through and pull out straws. So there's one of my alleles. I got too much in the frame, sorry. See if that makes a difference for you. So I have one of my alleles here. I'm going to take out another one. And so there's my first fish. So I'm hunting for fish. That's the first fish that's going to survive. So you can see in this, it says here, this initial round is a control. You're going to randomly pull out straws, choose your fish. No fish are eaten in this control round. For the remaining 3 generations in the round, the predators will eat every other blue fish. Alright, so colors of surviving fish. Alright, so in this case, I guess I should let you know, the predators discovered that the yellow allele made a sour tasting protein. So they're going to want to eat the blue ones. But only, so my control round, I'm going through. First one, I got a blue fish. So under colors of surviving fish, I'm going to now record one for blue. I'm going to go through and get 2 more, set those on the side. Red, and I got me another red. So this time I got myself a red fish. I'm using red as my yellow. So got myself a red fish. Okay. Going 2 more randomly. Oh, in this case I got a purple fish. And so I'm going through and I'm tallying all of my results. Okay, and under surviving alleles, how many of my blue ones do I have in the end? How many of my, in my case, red ones do I have in the end? When I go on to, so I'm going to tally all that. I'm going to then take all these, put them all back inside the bag after I've pulled every single one out and made my tallies for my control. I'm then going to go on to second generation. In second generation, this time I'm going to do the same thing. So I got a red with a red, so I've got a red fish there. That one, those ones survive because they prefer to eat the blue fish. Oh, in this case I got a blue fish. That's okay. That one survives because when you look at my instructions, it says the predators will eat every other blue fish. So I pull out 2 more. So I've got a purple fish. It survives. Got a purple fish again. It survives. And I'm going to trick you a second. I got another blue fish. Oh, this is my second blue fish. This one does not survive. So what I'm going to do is take this one out of my population. This one's been eaten. This one can no longer reproduce. So once I've gone through my second generation, what I'm going to do now is take all the surviving ones, put them back in the bag. Go through and do this again. I hope this helps. Like I said, I was confused the first few times I did this, or looked at it, but then when I actually sat down and did it last night, it made a lot more sense for me. So you're then just going to go through this entire document and do the same thing, and just keep track of the fact that in some cases you're going to have the blue fish are the preferred fish. In other cases, the yellow fish are going to be the ones that are going to be preferred. In some cases, it's going to be the thing that you're heterozygous, because we're dealing with incomplete dominance. Sometimes it's going to be those ones that are going to be selected for. So I'll work your way through it, complete the graphs and everything that goes with this. When you're finished, you can use the rest of the time to work on your study guides. All right. Hopefully that takes care of any questions you may have. I'm going to be home with my son, so if you have any questions, feel free to text me or send me an email. I'll have my email open on my computer as well. All right. Have a good day.